Drew, I want to give you the floor first, but what are your thoughts moving forward with the conservation going on in the United States for, whether that be at-risk species or if we want to call them like model or abundant species with, depending on your school and what you've seen or even what the Wild Times is doing, like what anybody's doing. I mean, where are you at with all these things?
Yeah, I'll touch on a few points just to kind of harp on something you're very much a supporter of. I definitely agree that you're right that that species of concern should not be, I guess, like a prerequisite of being taken account of. That abundance species are still just as valuable, and that we should be collecting all this inventory type data on them as well. Um, as you know, I'm a pretty big proponent of citizen science efforts, as you are, and I definitely think that's a great way to do it. Where we have people just going out and cataloging what they're seeing in terms of like an iNaturalist type of thing. But that's certainly there's certainly a role within there's certainly a role within the research community to do their due diligence as well. An example of where this has not been so much the case would be in the salamander world, especially in regard to hellbenders. I think we may have talked in the past about how throughout my hellbender work and through the people I've talked to, I've learned that hellbenders get like all the conservation dollars within salamanders because A, they're imperiled in a lot of places, but B, they're just so big and crazy and cool relative to a lot of the small. I mean, I shouldn't say cool relative, but it's so easy to like them because they're so big and they're so unique compared to everything else we have. Yet the Southeast is literally the salamander capital of the entire world. We have so many different species that have so much cool ecology, so much cool phylogenetic relationships going on, and all of them deserve some love too, right? And all of them need this love if we're going to have potential conservation challenges coming in the future. So that's something that I would start with to push for. Is the U.S. has a lot of resources to start to prioritize the more common species, and we should certainly go ahead and do that. And then the other thing you touched on was that model species side, which is something that's relevant to a lot of my research. I definitely think you're right that there's a lot of value in picking species that we have locally to us that we can kind of use to represent a larger proportion of herp diversity. And that's something I'm trying to do a lot with one of my projects, which is studying the mole salamander, which is Embystomous salpoindium is the scientific name. Ironically, they belong to the family of the mole salamanders, which gets confusing to describe to people. It's like, oh yeah, they're mole salamanders and they're a mole salamander. So that gets confusing. Um, yeah, so you can flash a picture of them up in your post editing, and then they're the type that's related to the spotted salamanders and the marbled salamanders, all the little dumpy guys, as I like to call them. But what's cool
They're
about
all
these
derp.
guys was that
They're all derps. That's what's fun about them.
exactly they're very much all derps in their own special ways. Um, but what's special about them in especially in the U.S., the only species that I know outside of the moles that do this is some populations of the tiger salamander, but they do what's called this facultative paedomorphosis, where you have your typical. The way I usually like to describe it to people is, you have if you're looking at a frog, you have your typical tadpole, which is the larval stage, which is aquatic, and then they transition to a terrestrial lifestyle as an adult frog. Everyone kind of knows that example. Salamanders, we have similar systems, although it's more variable. But what's special about this mole salamander is that it has this facultative paedomorphosis. So paedomorphosis means retaining larval characteristics into adulthood, and then facultative means not necessarily that it has the choice to undergo these changes, but that it can respond to environmental variables to do so. So in this case, we have this animal that starts out as uh, a larval individual, kind of like a tadpole, but with four legs like a salamander. And then, based on environmental cues, they have the ability to either leave the pond, like most salamanders do, where they'll suck in those gills through metamorphosis. They'll become a terrestrial animal, and they'll go leave the pond, and they'll only ever come back to breed. But then we have these paedomorphic individuals that retain these characteristics into adulthood. They sexually mature, they keep those characteristics, and they stay in the ponds for life unless they later do metamorphosis. So essentially, they look like an axolotl throughout life, just an adult salamander that has these gills and whatnot. Now, what's cool about this is the conservation implication here is that this species represents kind of all those different life history changes that amphibians do for the most part, right? Where you're starting out as, and this does kind of discount like direct developing salamanders that start out terrestrial. They're kind of the only outgroup here. But for the most part, we have the system where you can represent like the aquatic larva, you can represent aquatic adults, and you can represent terrestrial adults. And it's a perfect example of a model system where you can extrapolate these differences to a very high proportion of amphibian diversity, because most amphibians do some piece of that, right? They either start out as an aquatic larva and become terrestrial, like a lot of the salamanders that we have, or they start aquatic and they stay aquatic, like say a lot of our stream salamanders or our hellbenders, like I mentioned. So I think this is a perfect example of a model species, because it can be, you can put your or not your conservation, but your research funding toward that animal, and you can kind of extrapolate that, where you can take the findings that you get, you can take the data that you get, and you can potentially apply it all over the place, because it's representing multiple potential life histories. And we're specifically doing this with looking at the skin microbiome, which is something I might have described as a guest of yours before. But essentially, humans have the gut microbiome, right? We're kind of learning more about this in humans, where you have this gut bacteria that live inside of you, and they help you with digestion. They're very important to human health. Amphibians have that too, and while they have a gut microbiome, they also have a skin microbiome on that moist surface of the skin, which is also very important for health. Interestingly enough, it's been found that it can have significant ramifications for chytrid, which is something that we've been talking about. So what I'm kind of trying to do is see how these life stages are interacting with the microbiome, specifically seeing if there's certain microbial communities that are found in different life stages that are more able to combat chytrid, for example. And we can use that to see how these things might change as a salamander sexually matures, or as it goes from an aquatic to a terrestrial lifestyle. And we can use this to kind of hopefully start to make some conservation action toward a lot of different species, because we can see how in this model system that is changing.
How much is this microbiome like species specific and not like region specific? I guess that's kind of picking it up as it goes along. Would there be the potential, if you can find some combination that really helps to confer resistance against chytrid through like sort of, I believe I've read that it's like the microbes kind of help to fight off the fungus or something like that, just kind of incidentally, or there's some mechanism behind it.
Exactly.
Like that there would be a way to have a combination that works really, really well for that, and then prepare some sort of an inoculation to pass on to other populations, other species, kind of like how, I don't know, sort of a gross example, but like how they'll give people like a fecal transplant if they've had some kind of issue where their gut microbiome has been completely knocked out, to repopulate that so it can function again, like an inoculation of the biome to add to the microbiome on the skin of other individuals.
Yeah, you're hitting the nail on the head. I
Great.
don't believe it's been put into practice yet, but I know that a lot of people have proposed that you, similar to what you're talking about, that you create some sort of a microbe-rich therapeutic bath that you can inoculate animals with, and then kind of establish those particularly healthy microbes in a field setting. Because, like you said, that's exactly what's going on. There's a database and some prior literature that gives some suggestions of particular microbes that have been found to directly inhibit the spread of that fungus by being able to fight it off. And that is the way that you would do it in practice. I do worry about that method, because it kind of rings on the same, it kind of barks up the same tree to me as like introducing an invasive animal to fight off some other either invasive animal, or to like change some condition of an environment. And as we know, that never ends well. So I think it's certainly something that needs a lot more. I think we need to be very, very certain that this is a smart conservation technique. But that is, in the ideal world, how that would shake out exactly.
If I remember correctly, it's at least with like golden frogs. I want to say it was with like the yellow-legged toad or whatever, or yellow-legged frog. I can't remember which. But they were finding that like chytrid didn't affect the egg, and it didn't affect the tadpole. It was only when it got to froglet stage. And they were thinking a lot of it had to do with the protective layers that the animal already had onto it. And when you were talking about inoculation baths, like, we can treat chytrid in captivity, like, but it's this constant bathing with this. When I was working down in Panama, one of the things that they were talking about that what they want to do was basically creating an antifungal that they could add to the environment. But like you said, it's like inviting in an invasive species. There are so many hundreds of thousands, not millions, of funguses out there that are beneficial. And at some point, chytrid has a benefit, but it just didn't originate in most of the areas that it's from. That if you were to put out like some type of antifungal for it, well, then what are you doing to the rest of the environment? I mean, you could be looking at the complete destruction of an environment just from trying to take care of like one pathogen.
Yeah.
What kind of antifungal would this be? Are they talking about like a chemical or a bacterium, or to be determined?
Well, the last I knew of it, and it was more in the beginning stages, was that kind of like how here in Michigan we are eradicating the sea lamprey by attracting them with pheromones and can get them on the track. That was based off of genetic testing just on that lamprey and breaking down its genome. So last I knew, what they were trying to do is break down chytrid's actual genome, but funguses have been here probably longer than most animals have, and trying to break that down into like, being able to just direct at that one particular species is, I mean, that's like shooting in the dark. I mean, it's going to take years of research and funding to get to that point. But that may be the route that we end up having to take, because so far inoculation techniques haven't really been working all that well. Last I knew, they were trying to basically see if they could breed particular individuals that they had treated with for chytrid to see if they can pass on any type of immunities for it. But even that's just kind of like a really hit or miss thing at this point.
So from the genetic basis,
why would that pass it on? Isn't that kind of a really Lamarckian sort of concept? I thought that the main treatment was usually just like an antifungal bath, isn't it? Or is there some other?
So the main treatment is a type of antifungal bath, but if especially for like looking at the golden frog in particular, they're extinct in the wild, but chytrid is still active in the wild. So if you're going to bring this population back out, you have to make sure that they're going to survive it, and then that their offspring are somehow going to survive it. Because what's the point of breeding 10,000 or 20,000 of them to throw them back out there and have them all die within a month?
Yeah.
I mean, so it's a multifaceted approach. And
did you see?
Oh,
I'm sorry. I was just gonna say, did you see what National Geographic had out? This was like, I mean, this has been like 8 or more years, but they had on there, I don't know what species it was, but they were inoculating them, and they were putting them back out into the wild actually, because their offspring were able to survive and their offspring were able to produce whatever they were getting inoculated with. I don't remember it to a T. I just remember watching it. There was some research lab out there that was doing something very similar to what we're talking about. I'll have to see if I can find it and I'll send it to you guys, but it was really interesting.
Awesome.
I know that our species in the United States are very tolerant of chytrid, the majority of them, I should say.
That idea you're talking about, Darren, it sounds like it might be something that would work better with maybe like Dendrobatidae, because they can do the thing where they kind of sequester and repurpose and concentrate up toxins from like their food that they're eating out in the environment. If there was some kind of like maybe an oral thing that they could be given that would then be coming out on their skin or something like that.
Yeah, I'm not exactly sure. I could see if I can pull it up here while we're talking, but no.
Yeah, that'd be cool.
No promises for that.
Drew, the interesting thing too is like, especially with mole salamanders, being that I think like throughout the community that we don't really know a lot about a lot of mole salamanders. I mean, for an animal that's spending a good majority
quite clear in my research.
Yeah, I mean, like they're either in breeding ponds or they're underground, or we're not seeing them. Like here in Michigan, like I've worked with Frog Watch USA. I also worked with them when I was in Florida, and then like in our state we have Herp Atlas, and it's kind of what you're talking about. Is it's a citizen science program to go out and see how many we have, but I have only seen one tiger salamander in my life. I have never seen a marbled salamander, but I have seen thousands of blue spotteds and thousands of redbacks. But does that mean that, the marbleds and the tigers are out there? No, like we know they are. I have people contact me every year like, "Hey, I found 15 of them like out at this park." It's like, well, I'm looking for them. How am I not finding them? But like when you have a reclusive animal like this, like it raises a lot of questions about how do you effectively do the counts on them, and then when they do have this invasive pathogen, how are we going to attack it for an animal that we never really see?
Absolutely. Actually, kind of as a little bit of an offshoot, that makes me think of a video that's really going around the internet, going viral. I don't know where it was, but there's a guy and he's just draining out his pool, and he has it totally drained out, and he's down there with the the typical black pond liner, and he peels back with this fold in the liner, and there's like, I kid you not, like 300 spotted salamanders just all packed into it because of, like he said, like the guy even says in the video, they have that site fidelity, so presumably they were all hatched in that pond and did really well because it was man-made pond, probably not a lot of predation. All those salamanders go out and then they came right back. So like I said, you could go an entire year and not see any of them because they're all fossorial, just doing their thing. But then you get that right situation where you're in the right place in the right time, and you find literally 300 of them in a space that is like, the size of a desk chair or something, like not even super dense.
When I worked for Binder Park Zoo, I did all of our amphibian like surveying, and I would just keep going out throughout the year and see what I could find. And it amazed me how like one year we would go out and find 10 salamanders total, right? And then all of a sudden you go out a month later and you're finding 5 different species and hundreds of them. And it's a variance. I mean, you most people think of like, oh, I'm gonna go out and look for animals on a nice day. It's like, well, if you're looking for salamanders, that's actually the opposite. Go out on a crappy, semi-cold, rainy day, and you're just gonna find them everywhere. They're not gonna be out on, you're like, hey, it's 75 degrees out. It's like, no, they don't like it. So like some of that, like the numbers thing is, when are people looking? Because we want to do it when it's convenient as a mammal to go out and be looking for these animals, when really it's like we have this nature park not far from here in Hastings called Pierce Cedar Creek Institute, and it's something like 300 or 400 acres of reclaimed farmland, and they've recorded almost every single native species of herp in Michigan at this place. And so they advertise this thing they call the salamander run, where it's like usually mid to late March, they go out and they have videos of literally just hundreds, if not thousands of salamanders climbing over top of each other, going to breeding pools. And they find marbleds and tigers and spotteds and everything out there. And they're like, oh yeah, we just, we flip over a log, we find whatever species you're looking for. And I was like, well, that's amazing considering like here in my area, we just don't really find a lot of those guys.
For sure.
It really leads into a lot of the stuff that I'm doing, especially with like citizen science and also outreach. Ironically, one of the most common species that is in Pennsylvania and probably in Michigan is the spotted salamander. And there's a county that's not far away from me. That's one of the counties that PA Woods and Forests services. The spotted salamander has actually been declining for a couple of years now, and they're starting to realize that this could become a reality where they could lose it in almost all of the county. And ironically, last year, I had the chance to observe a vernal pool where the spotted salamanders were mating, and there were already offspring in there from last year, or from a couple months prior. So I found, from the help of people that I worked with out at the land trust, I found a population of spotted salamanders that double clutches. Now they didn't do it this year. At least I didn't see the spotted salamanders in there, but that is significant because it's a location basically in a suburban neighborhood that leads off into a forest, but also goes into a city. It's like really in a weird spot. But there's habitat out there for them. And now, because they're declining, there might be a shot to get them protected. But it'd be really interesting to see, I guess, where this population compares to some of the other ones. And Drew, you'll have the chance to see all this stuff because you're coming up next week. But it's just something to think about, like especially what you're saying, Sean. A lot of people want to go out and look for these animals whenever it's convenient, and if they don't find it, we could even take it a step further and say they declare them extinct, or they say they're no longer here or whatever. But a lot of the work that I'm trying to do is find these animals that have been, at least in Pennsylvania, like given up on, or like they've never been documented in certain locations. And there's a lot of different reasons why. I mean, we could take it as, like I said, we're creating a database for the model and abundant species. So that way, if something happens, we're in place to go and save or help that animal in one way or another. But also to potentially produce outreach and produce like people having that opportunity to care about something that nobody even knew was there. I mean, that was the example for the gray tree frog in 2 counties, and hopefully for other species. But for the conservation work that I'm trying to do, I'm trying to take it even in a step further by dividing it in different phases. You know, phase one is just like learning about the animal and understanding where it might live. Phase two would be trying to locate breeding populations. Phase three is actually trying to do road rescues and monitor that population on a yearly basis to see how many individuals they have. Year four is actually looking to create, I'm sorry, phase four is trying to create habitat, translocation, and all these other things. And I mean, these are some of the conservation methods I think that are going to be used and they're needed for a lot of our species. And I know they're being applied in other countries, but just something to, you know, to think about with going out herping and stuff like that. I know for me at least, like I was just saying, I'm definitely not like, it's kind of weird. I have these conversations with friends. They're like, "Oh yeah, we're gonna go herping." It's like I wouldn't necessarily consider myself a herper because I'm not trying to travel the globe to just say I found the salamander just to find it. Like I'm really trying to find something to actually help manage it or do something that I can with it. And you'll see that, Drew, when you get here. But like, it's a little different of a perspective than like some of the herpers, at least in the state of Pennsylvania. I'm not going to say like in Michigan, but like I'm definitely not just out there herping to go herping.
Right. Well, I mean, there was an instance where I was talking to the guy who founded the Herp Atlas here in Michigan, and we were just talking about different salamanders we found. And I'm originally from the Upper Peninsula, and I was like, "Yeah, dude," I was like, "I used to find blue spotteds all the time." He goes, "No, they're not in the UP." I go, "Oh no, they are. Like, I can give you an exact address because they were all over my backyard. I used to catch them by the dozens when I was a kid." And he is like, "Well, input it in Herp Atlas then." He goes, "And we'll send a team up there and we'll see if we can get permission to go on that land and look for them." He goes, "But this is the whole purpose of having something like Herp Atlas, a citizen science program." He goes, "We would have never thought to look in the, east end of the UP, which is mainly like rocks and like basically like fir pine forest." He's like, "That's not really habitat." And he goes, "Really, like, you only have a couple nice months out of the year." And I'm like, "Well, my backyard had like a bunch of vernal pools and was on the edge of a forest." I'm like, "But I would flip over logs all the time and I would find blue spotteds. I would find green snakes, copper bellies. I mean, pine snakes, garter snakes. I mean, we had all kinds of stuff." And, I was 3 miles from Lake Michigan, and one of the colder parts of the UP. So, I mean, we need these programs and we need people to go out and actively look to help us out from the conservation aspect because we don't know what's out there. We don't know where they're at, but we do know that if people go out and help, that we're going to find populations we didn't know existed.